nitrate, a metallic salt. The cells are fixed with Fontana's fixative. Then they are treated with mordant so that it the staining which takes place at a later stage is done very appropriately. And finally, ammoniacal silver nitrate, when it is being used and the heat is applied, ammoniacal silver nitrate gets oxidized. As a result, silver oxides are formed, which are blackish brown in color. Due to the formation of insoluble precipitates, this helps to increase the apparent diameter of the cells. So spirochetes being too thin, their diameter is increased. As a result, visualization also is easy. So this is the method which is being specifically used for spirochete staining, and that method is known as a Fontana's silver impregnation method. Apart from that, we have certain special staining procedures for spirochetes, then for staining of mycoplasmas, rickettsias, fungi, protozoas, and all other different kinds of microorganisms. Okay, since we do not have much time, we will not be going into the details of various cytological stainings. We will just have a glance. Cytological structures like endospores, flagella, capsules, and cell walls, they are also stained like methods which we commonly use. Dyer's method for cell wall staining, in which we use cetyl pyridinium chloride. Then we have a flagella staining, in which Lifson's and Loeffler's methods are used, where the flagella are used because the stain precipitates along with the mordants on the flagella. Otherwise, the flagella are too thin. It is beyond the capacity of even a normal compound microscope to visualize flagella unless and until the diameter is increased. So this is how we stain flagella. Then we have capsule. Capsules can be stained either by negative staining, by nigrosin or India ink, or it can be stained by using another kind of dye, that is Congo red, in manual staining method. Then we have certain other staining like spore stainings, where basically spore being a resistant structure, application of heat, that is the intensifier, and a very thick, very strong dye like ZNCF, it is being used. Then we have certain staining of nucleus or metachromatic granule stainings. These staining techniques are normally done by the students at the graduation level, and they will be discussed during the practicals by the respective teachers. At the end, I would like to thank the BISEC studio crew, the staff members of the BISEC, for their excellent cooperation and presentation of the subject. I would also like to thank on my and on behalf of Madam to the Commissioner, Department of Higher Education, for giving us the opportunity to reach to the students and to give them certain aspects of microbiology. Thank you very much.
Thank you.